Okay, welcome back, guys. So if you're here again, you obviously still don't know what the fuck we're doing. And now this is even more shameful because what we learned was from 7th and ninth grade biology. Obviously, you should still be ashamed of yourself. But there's hope for this goddamn situation you're in. I'm gonna reteach you the damn basics of the cell cycle. And leave comments if you don't understand, and maybe I'll make another video explaining it if I feel like it. Okay, so once again, I'm just going to go through slides that have been made for the class in general and explain what's going on them, and I'll probably also go to outside sources because this does not probably have everything that I want to talk about. Okay, so the cell cycle. Why do we give a damn about the cell cycle? Well, the cell cycle is important because it's what allows the cell to continue on. Like, we need more cell. We need to keep making cells if we're going to keep living. Cells aren't immortal. They eventually die off. They have errors and all so forth. Like, those kind of errors build up if we don't keep dividing. So eventually we need to make more cells. We can't just stay in a stable number. And that's the reason why we have the cell cycle. To go from having one cell to eventually going to the end with mitosis, which then creates our next two cells that still maintain their still maintain the same genes as before. Okay, so what we probably want to first talk about is what the mitotic phase is, which is... The mitotic phase is basically the very short phrase in this very short phrase right over here. It's over here. As you can see, it only takes up a very small portion of the time. It's composed of cytokinesis, which is when the cytoplasm, see cytokinesis, cytoplasm, yeah. That's when that breaks apart, and mitosis. And mitosis is the part where all the DNA and whatnot forms together and then breaks apart at the end. That's where mitosis part is. So the mitotic phase are those two parts. As you can see, it's a very short part, and that produces the two cells. And then there's the long interphase. This is the interphase. It's composed of like the G1, S, and G2 part, and this accounts for majority of the cell's time, about 90%. Okay, this picture is really ugly, so I'm just going to look some stuff up. Okay, so first what we probably want to go through is the cell cycle, ignoring the mitotic phase. We're just going to talk about the interphase, yeah. We're going to be talking about the interphase portion of the cell cycle. And I'll try finding a picture of it. Okay, they don't have a nice picture of it. That's cool. God damn it. Where is the goddamn image of the damn... God fucking damn it. Okay, fine, whatever. It doesn't really matter. We'll just go with something like this. Okay, so the interphase is basically everything that's going on before the mitotic phase is happening. So the G1 phase, or also known as the gap 1. So that's just basically the phase where our cells are just conducting what they're supposed to do. They're just growing and doing what they need to do. Like, this is the point where the nerve cell would be conducting impulses. This is where all the cells are mainly at initially. It's just conducting what they need to do. Then there's the S phase. The S phase is where DNA replication starts and where all the DNA is complete. Like, by the time the S phase is completed, DNA replication is done. Alrighty then. And then we get to the G2 phase, which is the point where, what is it? The cell begins preparations to enter cell division, which obviously is the mitotic phase. So basically the G2 is just the preparation stage for the mitotic phase. Okay, so that is interphase. It's just basically what the cell conducting what it needs to do as a cell, and then eventually it gets to the point where it begins cell division. And now we get to the important part, which is probably where the majority of the thing is. We're going to be talking about the mitotic phase. And that's probably the more important part. And if I'm correct, there was a very nice image inside the slides with this. Why am I doing this? Anyways, okay. So we'll just go on to the next slide. Wait, never mind. Let's first go back a bit. So with the DNA itself, the DNA, while it's in the interphase, is actually like very loosely bound. Like it just looks like a clump. Chromatid. Oh, whoops. No. I'm not trying to look for chromatid. Oh my god. What is it? Where's a picture of that? Okay, maybe we want to look at a G2 phase of it. This is really not working out right now because I don't know where the fuck is. Oh, here we go. That's a pretty nice picture. Anyways, as you can see, like this picture. Yeah, as you can see in this picture, the DNA is kind of spread out. It's in the nucleus and it's just spread out. There's no organization. It would be very hard to see. In fact, I don't think it's possible to see with a conventional light microscope. So yeah. But then as we go through the mitotic phases, it begins to coalesce. It begins to come together. And then it forms this familiar shape. You should recognize this. It creates the chromosomes that we all see during the DNA replication. So at this little joint here, there's a centromere. It's called the centromere because it's like at the center, and it has, this is where a lot of the things will like connect to. Later on, I think they're called, what is it called? Oh yeah, the kinetochore microtubulars will connect here, and that's important later on. That is what's going to allow them to be pulled apart. So, this is the centromere, and then this is the centrosome arm, which is basically the longer portion. And then it will duplicate itself, making two of them. And eventually they'll be separated into two. And that's the basic idea, but then you want to be in more detail over the entire cell. So, here's a picture of the G2 phase. Oh, here we go. Look, there you go. All the chromatid is just spread out. It's duplicated, like it's already been duplicated since the S phase, but it's just spread out. Also, important thing to note, the centrosomes have also duplicated. There used to be only one centrosome, and the centrosome is just basically the area where all the microtubulars are commanded from. It's like, it organizes all the microtubulars and filaments, if I'm correct as well, which you should know are what give the cell rigidity and whatnot, and structure. And you should also know what they're made of, which I've said before. They're made of tubulin. Okay, and then we are probably going to want to talk about the first in general, the 4 main stages, and then there's also something called the pro-metaphase, which is like the more interesting thing. Okay, let me just get a Word doc and then just show you what's important. Goddamn. Okay, so what's important right now when we're talking about the mitotic cycle is probably the first thing. So it goes from, obviously, the G2 phase. It goes from G2, and that is just all the preparation stuff. That's just the preparation stuff, and I might have misspelled it. I don't really care. I really don't care. Yep, I spelled that wrong. Okay then, so that goes from the preparation stuff, and then we enter Metaphase. I'll go into more detail later on. So currently I'm just going to give you all the names. There's Metaphase, this pro-metaphase. There's... Oh, wait. No. Ignore that. I'm retarded. That's retarded. First, there's prophase. Then there's pro-metaphase. Then there's metaphase. Then there's anaphase. And then there's telophase. And then sometimes people consider cytokinesis part of it. I don't really know if you... It's like, this isn't really related to the DNA stuff, so it's arguable whether you include it or not. It's important, but biologists like to argue whether that crap is should be part of it. Oh, obviously this is not part of it. This is part of interphase. That's just important. That's the phase before prophase. Okay then. So if you need a mnemonic to memorize them, this mnemonic is for PMAT. And it fails to take into consideration the pro-metaphase, but you should know that basically there's a phase in between prophase and metaphase, and it's a combination of the prophase, you see pro, and then metaphase with the meta. Pro-metaphase. And you can remember that it's called pro-metaphase because pro comes first, meta comes later, so pro comes first, meta comes later. And so basically the four main things are prophase, metaphase, anaphase, telophase. And what I learned from anatomy is basically a mnemonic that is as wrong as possible will make you remember it for the rest of your life. So, here is a mnemonic for you to remember this. You can say, porn makes anyone try. Okay, remember, porn makes anyone try. You'll never forget that if you remember this. You'll always remember there's PMAT, which is porn makes anyone try, prophase, metaphase, anaphase, telophase. It's pretty wrong, but you must admit, it's going to scar you for the rest of your life, and you will never forget biology again. Okay then. So that is basically the overview of this crap, and now let's go into detail about what they do. Whoops, that's the wrong thing. Okay. So we have prophase, and prophase is like the initial part. The chromosomes have assembled, and they make the two sister chromatids, as you can see, and then they have the centromeres. They've assembled together. The nucleus has still not degenerated, it hasn't been broken down yet, but the nuclei has disappeared. Notice or nucleolus, if there's only one, but if there's more than one, it would be called the nuclei. Notice that it's disappeared, they don't have it anymore. It's gone. And then, yeah. So, and then also, note the spindle, the centrosome. It's begun to create the mitotic spindle fibers, and these kinds of things that are pointed out of them are called asters. Because aster, star, and this kind of looks like a star, so they called it asters. Don't ask me why people do this. Okay, and then we go on to pro-metaphase. So, this is the phase between it. Basically, the cell wall nuclear envelope has broken down. The spindle fibers have begun to interact with the chromosomes. And they interact... what is it? They interact with the it's kinetochrome. The kinetochore, which is a specialized protein structure basically on the centromeres. And basically, the spindle fibers attach to them over here. Not all the spindle fibers will attach though. And yeah, oh right, these are made of microtubulars. These microtubulars attach here, and they're called the kinetochores. And then there's the non-kinetochore microtubulars. They're basically the ones that don't attach to the centromeres. You see, they don't attach. Cool. And yeah, they begin assembling, but it's pretty disorganized right now. That's what pro-metaphase is. Then we get to metaphase. Now you see they've all lined up. They lined up into this nice plane. Right. Something important to remember. Although on pictures it looks like a 2D, so it looks like a line. It's actually more like a plane, like there's a plane because cells are three-dimensional, just remember that. Note this metaphase plate. There's not an actual physical plane, they just call it the plate because it's basically where they all line up. It's this imaginary thing that biologists did because it makes sense. It's kind of like Earth. We put the equator even though we don't really need the equator because it's not really there, but we just put it there to make it easier for us to understand. Okay, the spindle fibers have tugged them towards the center and now they're all lined up quite nicely. And yeah, that's basically metaphase. They all line up. And then we have anaphase. This is a pretty cool phase. This is when they actually start breaking apart. They get basically torn apart, but not really torn apart. What happens is there's actually an enzyme that cuts them apart. If I'm correct, the enzyme is called separase? Is that what it's called? Don't quote me on that. I'm not actually sure if that's what the... But anyway, there's an enzyme. It cuts it apart and basically they can start being taken away towards the end. And what's kind of important is note the non-kinetochore filaments. They've actually gotten longer and they're still interacting with each other and they're actually kind of pulling the cell apart. It's becoming larger and it's being pulled apart right there. That's important because that's going to help us get to cytokinesis in the telophase part. So they're being pulled apart and then they're being pulled on in. There's actually a couple different ways they get pulled on in. We'll talk about those later. But anyways, anaphase is just basically a phase where they start getting pulled apart. And then we have telophase. This is basically the point where the nucleus starts reforming and basically the cell begins to reform. The nuclear envelope from those old fragments earlier, they get pulled together. They put them together and start making new nucleuses. And the chromosomes start to break apart. They start reverting back to what they originally started out, which was this unfurrowed mess that we call our DNA. And then we also have cytokinesis. Cytokinesis is interesting because basically in animal cells we have a cleavage furrow, which is basically actin and myosin, which is also found in muscle if you didn't know that. Basically it starts contracting on itself slowly at the center and it starts pulling them apart. They contract where the metaphase plate was, which is pretty important. So they kind of break it apart into even parts. Okay, then what is this? Assembly of the microtubules started in the centrosomes. That is true. They do start in the centrosome. Basically, okay, the centrosome was, as we said earlier, this part, and basically spindle fibers come to it, and they also interact with these things, and they make this beautiful thing with the spindle fibers that are connected to the centrioles. Oh, one little thing. You don't actually need centrioles for cells to divide. You could destroy the centriole and it would still divide. The centriole helps aid this process, but they're not actually required for the process of cell mitosis. Okay, so yeah, the chromosome kinetochore is captured by the microtubules. Basically, the proteins on the centrosome, they get pulled by the microtubulars, and then they get pulled towards the poles, basically. And then there's a tug of war between the ones from the top and the ones from the bottom, or if this cell was rotating 9 degrees from the left and the right, it really doesn't matter what. Anyways, there's a tug of war. The tug of war, no one wins, and they actually settle in the middle, on the metaphase plane, as we saw earlier. Cool. This is what I was talking about. There's multiple hypotheses on what actually moves the chromosome along. They're actually all right. Quite a few of the hypotheses, they're all right. One of them is the one where, the motor protein walks along the microtubular, basically, and as it walks along, it also breaks down the tubulin apart, and once it gets to the end, obviously, there's nothing to break apart, and it stops moving. But there's also another theory where they're getting reeled in. Like the centrosome reels in the microtubular, kind of like a fish. It starts reeling it in. Those are both actually correct. Both of them happen in cells. It's just like, depending on what type of cell, one is preferred over the other one. But both of these hypotheses are correct. Okay, and here we're talking about the non-kinetochore microtubulars. And yeah, they're responsible for lengthening the cell on the axis, as I said earlier. Basically, they get longer and they start kind of tugging on the cell, forcing it apart. Yeah. Okay. Okay, and then we have, this is important, cytokinesis. Basically, I was talking about earlier, there's actin and myosin filaments around on this, where the metaphase plate was, and it starts breaking, it starts polishing on the cell, and eventually the cell just breaks apart into two. Okay, and then we have to talk about cytokinesis in plants. Cytokinesis in plants are very different. They do not do what the animals did, which was have this cleavage furrow. What they do instead is they actually just build up a new wall in between the two. They just like build a new wall. So, as you can see, this picture is actually very unclear about what's going on. Here you go. This is much better. So basically, the Golgi body makes some vesicles. These are the two vesicles. They get sent towards the middle of the current cell. Those vesicles contain materials that are required to make a plasma membrane in between the two of the cells and a cell wall. If I'm correct, they create cell wall in between, and basically they build up the cell wall. They keep building it up, and then eventually the cell wall is completed. Membrane gets placed over it, and look, now we have two plant cells that are right next to each other, but are still connected to each other because of this new plate in between them. And that's the reason why plant cells are very rigid, because as you can see, they're all connected. Okay, and this would be a picture of an actual cell based on dyes. So basically, as you can see, interphase. Can't really tell what the hell is going on. It's just like this purple glob. Then in prophase, you begin to see that there are assembling together. You can kind of see there's lines here, sort of. It's like you can sort of see the chromosome. I mean, it's not a very clear shot, but you can sort of see the chromosomes start developing. Then we get to the pro-metaphase. Basically, this is the intermediate phase between these two. They haven't lined up. They're starting to interact with these fibers, the spindle fibers, but you can't actually see them in this dye. The dye wasn't designed to pick those up. So basically, yeah, it's just a globular mess. Then we get to metaphase. As you can see, it's pretty organized now. Now it's pretty lined up, and yeah, it's pretty lined up. Then we get to anaphase. Yes, you can see them being pulled apart. Cool. And then we get to telophase, where they're basically, the two cells are really pulling apart, like also cytokinesis happening, and you can do themselves. Cool. Okay, and this would be the ideas of what evolution might have seen from that went from being like a single cell prokaryotic cell to the eukaryotic cells we see now. And prokaryotes reproduce by a totally different way. It's called binary fission. It's like a sort of kind of like mitosis, but it's kind of not. Like, it's like basically they have this place called the origin of replication. It's where the replication starts for your cell. Oh right, important thing, they don't have a nucleus. They just have a mess of chromosome everywhere. It's just a mess, and they don't really organize it. They keep it as this globular mess, basically. So they have this origin of replication. That's where it begins replicating, and then the two of them eventually go through this entire mess, replicating everything, and now they have two copies. The two origins, they pull apart. They like go to different poles, like kind of like the mitosis, and that's where they kind of get the idea that mitosis might have evolved out of this, because you see they also form two poles. And then basically they get pulled apart, and I mean, they get pulled apart, and new plasma membrane grows in between the two, and then they become $2 cells. Cool. And here's some ways of how it's do, as we saw how bacteria did it earlier. There's also dinoflagellates. Can't really pronounce that. I'm sorry if I butchered that really badly, but I really don't give a fuck that I butchered it, because guess what? You probably will butcher it too, so fuck you, anyone that's trying to correct me. Anyways, this one is interesting because they don't break down the nuclear envelope. They basically keep the nuclear envelope, and they kind of do the same thing as the bacterial chromosome, except they have a nuclear envelope. They pull apart inside, and they make 2 cells. Cool. Then we have diatoms and yeasts. They also keep the nuclear envelope, but it's basically kind of like the eukaryotic, but basically they do the same thing, but without destroying their nuclear envelope. Kind of similar. As you can see, it's sort of similar. It's whatever. Then we have the most eukaryotic, which is what we were talking about for a long time earlier. Okay, we will talk about this in the next video. This will be cell cycle control system. This is basically how the cell keeps itself from doing it too early, blah blah blah, like not fucking itself up. Yeah, we'll talk about this in the next video. Alright, so leave comments below if you need help, and then yep, that should be good for cell cycle. If you don't understand something, just leave a comment.